So before we get started, I just want to let you know that the products that I'll be showing today are intended for molecular biology applications only. These are not intended for the diagnostic, prevention, or treatment of a disease. Now, just to make sure that you guys are aware of the different features that are available for today's webinar, if you guys have any questions, please go ahead and utilize that Q&A box that's located at the bottom of your screen. This is going to help us keep track of what questions are being asked. And it's also going to help us keep track of what questions are being answered. So you'll see that icon somewhere in the bottom of your screen. Now, we do try to make this interactive to get a feel for, whether we're showing the right stuff that you guys are interested in. So you'll also see at the bottom of your screen this raise hand icon. So you guys can go ahead and just click on it just to make sure that it is working for you guys that are attending today. That would be awesome. And so that raised hand is located at the bottom of your screen. So you can see that many of you are able to locate that icon. Great. And then if you do experience any technical difficulties, let us know through chat, and we'll try to help you as best as we can. So today we're going to start off with just a brief introduction to the OmicsSoft land, just so that you guys can get a feel for what it is and what it does. And then we'll go in more in depth and see how you can use the GSLand to explore public data sets. So here we're going to show you how to select different comparisons of interest and identify potential biomarkers by taking a look at how you can generate some volcano plots. We'll then take a look at how you can leverage public expression data, allow you to identify how your genes that may be of interest are expressed across different conditions through the use of multi-gene heat maps, and then how you can also generate some box plots as well. Now, because we do have a wide variety of different softwares for different types of applications, we'll take a look at how you can then take the information that you obtain from OmicsSoft, and you can use the human gene mutation database to identify variants for those genes that you identify within the OmicsSoft lands. And then we'll take a look at how you can further expand and discover potential mechanisms using engineering path analysis, or IPA. And then lastly, today we are going to be focusing on only the GUI or the web versions of OmicsSoft, IPA, and the human mutation database. But do know that there's different ways that you can access the data, and we'll take a look at those different ways towards the end. So users like you want to be able to explore publicly available data for biomarker discovery, target validation, or to look for additional genes of interest. And so in OmicsSoft, you can access comparison data for your gene and disease of interest and use the comparison details to identify public data sets that you can then explore. You can then use different comparisons of interest to generate volcano plots where you can dive into what are the potential upregulated genes, what are the potential downregulated genes that you can use for biomarker discovery or for target validation. Now, in OmicsSoft, you can generate a heat map for differentially expressed genes. And so here you have a heat map of comparisons. So here we're taking a look at disease versus normal comparison for Alzheimer's data sets. And over here on the y-axis, we have different genes that you are interested in, are potential biomarkers that have been identified in different publications. And so here, the red coloring is going to represent a upregulation in disease versus normal, and then the green represents downregulation in disease versus normal. So taking a look at this heat map, you can quickly identify, for example, that mediator 13 seems to be upregulated in Alzheimer's disease versus normal control across these different tissues that you see here. And then you can see that although MED13 is upregulated, we can see that for the most part, GNB5 tends to be downregulated across all these different tissues in Alzheimer's patients. And so by quickly looking at this heat map, you're able to identify what could be some key genes of interest. In addition to taking a look at comparison data, users like you generate a like to generate sample level gene expression heat maps, where you can see, for example, right, that if you take a look at MED13, you can see that for the most part, it is has higher expression in Alzheimer's disease samples compared to the normal controls. Again, we're here, the red represents a increase in expression and the green represents a decrease in expression. And then if we take a look at GNB5, right, we can see that for the most part, you see sort of like a lower expression of this gene in the disease samples compared to the control samples. And so again, visualizing the heat map allows you to take a look at how your genes of interest are expressed across different individual samples that you may be interested in. Now, rather than looking at heat maps, right, because sometimes they can get a little messy, you can generate box plots. And so here you can see that we have a box plot for each of your individual genes. And then here, each individual dot is going to represent a sample that is subgrouped between normal control and our disease state, which in this case is Alzheimer's disease. And so as you can see here, many of these markers are differently expressed between normal control versus Alzheimer's disease, allowing you to validate these potential biomarkers. You can even get some p-values and sort the graphs based on p-values to prioritize your targets that you may be interested in. Now, often users like you are interested in identifying variants in genes or targets of interest. And so with the Human Gene Mutation Database, or HGMD, you can take a look for different variants that may be identified in a disease, that might be identified through publications, and see how those particular variants are tied to a particular disease state or phenotype, get information on whether those variants are classified as disease-causing variants, and then dive deeply into the biomedical properties of these variants to see how deleterious they are, or whether these mutations have a gain or a loss of function. Now, users like you can also leverage engineering path analysis, or IPA, to further explore potential mechanisms and validation. And so here, GNS is a marker that's been identified in publications. And so within IPA, you can build a network connecting this particular target of interest to Alzheimer's disease through various intermediates. And here you can use IPA's knowledge base to look for what are some of the known biomarkers or drug targets that have been used within different applications. You can easily identify them within your network. In addition to building a network, you can take a look at your gene and get further insights and see, what is the gene about? What is it a member of? And then what particular pathways this gene is involved in. Now, since all of these different softwares work hand in hand after identifying a single pathway that your target is involved in, you can go back into Omicsoft and query that entire signaling pathway to see what genes are up regulated in the disease state compared to the control samples, and then what genes are going to be downregulated in the disease state compared to the control samples, allowing you to identify, what other genes that are involved in this pathway could be potential biomarkers or potential targets that you can further assess. And so I'm going to go ahead and just launch a quick poll to see if these are some of the results, some of the visualizations that you're looking for when you're querying Omicsoft, or when you're querying HMD, or when you're using IPA. I'm going to go ahead and launch that poll. So if you guys can So you guys can go ahead and just fill that poll. That would be awesome. That's just going to give them some feedback in terms of, what we're showing is what you came here to see. So you can see that many of you actually like the illustrations that I've shown so far. So that's awesome. Now, first, we're going to start off by talking about Omicsoft. And so many of you may not be familiar with Omicsoft. Some of you may have used it before. And so our Omicsoft technology allows users like you to explore public data sets. And so we have a team of scientists that manually curates and analyzes public data sets that are deposited, for example, in GEO, SRA, that they may be from consortiums such as TCGA, and process that data and deposit it into this LAND database. And so they basically download and QC the raw data, process the data using a standardized pipeline, quantify it, they perform statistical analysis, and store all that information into our LAND database, allowing you to compare between data sets. And so this means that data sets in Omicsoft LANDs also need to pass a quality control matrix, giving you the confidence in the data curated and processed in the LANDs database. In addition to taking a look at the raw data, our curation team also performs quality control on the metadata that's associated with the data sets. And so after curating the metadata, these terms are then converted into control vocabulary that allows you to easily take a look at characteristics across these different data sets. And so here, the key advantage of this is that, if you were able to do this on your own, it would take a tremendous amount of time and resources, as opposed to just using this LAND platform where you have everything ready to start. And so the Omicsoft LANDs represent curated data from over 750,000 samples coming from the public domain that are going to be made available to you through the different LAND collections. And so today we are going to be focusing on disease LAND, which contains over 321,000 samples from hundreds of non-oncology diseases from individual studies, from GEO, SRA. It also includes over 161,000 samples from LINC's perpetuation studies. Now, for those of you that are interested in oncology, for example, OncoLAND contains more than 210,000 samples from individual studies such as GEO, SRA, and more than 38,000 samples from consortia such as TCGA, Target, just to give you some examples. In addition to these disease samples, you also have information from the body map collection that looks at expression in normal tissues from GTEx, Blueprint, the Human Protein Atlas, for example. And then we also have collections to take a look at single cell. So single cell LAND contains 18 million cells from thousands of samples, taking a look at anything from non-oncology to oncology. And so this just kind of highlights the broad spectrum of different types of data that you can find within the LANDs. And so let's go ahead and use disease LAND to identify and select public data sets that you can use for your biomarker discovery and target validation. Okay, so let's say that you read the single cell study where they used a mouse model of Alzheimer's disease to look at healthy and disease-associated astrocytes. Now, in this publication, the authors found GFAP expression in disease-associated astrocytes. And so in the disease-associated astrocytes, so we take a look at this chart right here, which represents here, cluster 4, there was an increase in this population, which has a high expression of GFAP. And you can also see that right here in this cluster 4. So you can see that cluster 4 has a has a higher average of expression of GFAP as well as the percentage of the cells expressing GFAP. And so using Alzheimer's disease and GFAP, let's go ahead and use public human data to look for potential biomarkers or targets. So let's go ahead and go into Omicsoft. So for those of you who are following along, I'm currently using Omicsoft Studio version 12.8. And some of you may be using different versions. So some of the default coloring or default views may be a little different than mine. So feel free to contact us or your IT administrator to see if you can upgrade to the latest version of Omicsoft. Now today I'll be focusing on the lands tab, which is right up here. But do note that we have other options available. So if you're interested in processing your own FASTQ files, using some of the pipelines that we use to process the public data, feel free to check out the analysis tab right here. And so to go ahead and select the land that you are interested in, you can just simply go ahead and use the option to, not that one, to select land. And so here I already have our land of interest that we're going to be using on display, but you can easily select lands by simply going to the select land and then selecting your land of interest. And so as I mentioned, we do have different types of collections that may be interested. So the body map collection is going to contain normal expression information. We have the cell line collection to look at different cancer cell lines. And then for today, we're going to be focusing on the disease land collection that has non-oncology studies for both human and mouse and rat. But if you guys are interested in oncology, we do have the OncoLand collection, which has different types of data from individual studies, which you can find in OncoHuman, to consortia information such as GTEx, such as TCJ, for example. And then we also have our single cell line collection for both human and mouse data. And so today, you're going to be focusing on human disease. So you can just simply click on the hyperlink, and it's going to open up this page that you see here. So when you open up human disease, the default view is the sample distribution view, where you have disease categories on the y-axis and then the number of samples on the x-axis. And so for those of you that are new to Omicsoft, the select view allows you to select how you want to view your data. Here, this panel on the left-hand side contains different filters based on the metadata that we curate that allow you to identify the data that you are interested in. And then here on the right tab under the task, you have the task tab that allows you to change the way that this center chart displays information. And then the legend lets you know what type of information is being displayed. And so today, the main areas that we're going to be using for the most part is going to be the select view to change how we're viewing the information, the left panel to filter for our data sets of interest, and then we're going to be using the task tab to sort of customize the way that we're viewing things. So today we're going to be focusing on GFAP because it was identified as a potential biomarker and it is highly regulated in disease-associated astrocytes. And so to begin, you just simply go into the search bar right up here and then type in your gene of interest. So let's go ahead and type in GFAP. And as you do so, it is going to start to populate. Well, may be having some issues, but there we go, it's a little slow. So once it starts to populate, you can just simply select your gene. And what this is going to do, this is going to query human disease and bring up the information relevant for your gene of interest. So here, the default view when you search for a gene of interest is going to be the comparison view. Comparing, in this case, your gene of interest, GFAP, in disease versus normal comparison. And so this chart shows different disease categories for the case on the y-axis, and then expression information on the x-axis. So here, every bubble is going to represent a different comparison. And if you hover over the bubble, you can get information on what that comparison is. So then looking at all of the different comparisons that are available for your gene of interest, you can filter down for your disease state by simply utilizing the filters that are available here in the left panel. So here you can filter down to Alzheimer's by simply going into the Projects tab. And then once you're in the Projects tab, you can open up the menu for the project information. And this is going to show different filters that are specific to project information. And then here, because we have a specific disease that we're interested in, you can go ahead and click on the little filter icon under disease and either search or select your disease of interest. So let's go ahead and start searching. So as you do so, you can select your disease of interest. So today we're just selecting one, but you can select multiple diseases that you may be interested in. And then going ahead and click OK. So here you can see that you now have a filter for Alzheimer's disease. And so this allows you to take a look at comparisons, which again are going to be represented by bubbles for samples from different Alzheimer's disease projects, where the disease state is being compared to normal. So positive log to fold change means that GFAP is upregulated in Alzheimer's versus normal control, and then negative log to fold change values mean that GFAP is downregulated in Alzheimer's disease versus normal control. So although the magnitude may be different, you can see that the majority of the public comparisons show a upregulation of GFAP in Alzheimer's disease versus normal control. Now here, as I mentioned, you can hover over the bubble and it will show some information on what those comparisons are, but you can get more precise details by simply just selecting those comparisons. So let's go ahead and draw a rectangle and just select these comparisons of interest that have a twofold increase. So you can just hold down your left mouse click to select those comparisons. And so when you do so, it is going to bring up that information in the form of a table here at the bottom. Right, so here you have now a table. So this table at the bottom allows you to take a look at the sample level expression for the comparison that you are interested in. It also allows you to take a look at the different metadata information for that project that generated that comparison. So here, for example, you can see that this is a disease versus normal comparison, and it gives you information on what that particular comparison is actually doing. So we're taking a look at a specific tissue, and it is disease versus normal. You can see that this is a microarray study by taking a look at the sample data mode, so it says expression intensity probes. And again, you can scroll down to get information on what are some of the metadata. And then right here under the comparisons box, you can see the four comparisons that you highlighted. So you can see that we have one comparison coming from this project, GSE 48350. And then you also have 3 comparisons coming from this other project, GSE 5281. And so this table provides you some information on what the different comparisons that you're interested in are, but you can take a look at more information and explore the differential expression that's associated with these comparisons by simply going to the option to browse selected comparisons. So when you click on the browse selected comparisons, it is going to bring up that information in the form of a volcano plot. So just a show of hands, how many of you use or like to use volcano plots to identify what are some genes that you can potentially target based on how they're differentially expressed? So I can see that some of you do use volcano plots to identify potential genes. That's great. And so here, as I mentioned, by clicking on the Browse Selected Comparisons, this is going to open volcano plots for each comparison that you selected, allowing you to identify genes that are either going to be upregulated or they're going to be downregulated. So let's go ahead and change the layout of this chart. So currently you have 4 charts, right? But you can change the layout to visualize them in a single view by simply just using this little drop-down menu right here. And here, let's go ahead and select the option 2 by 2. So now you have your 4 charts displayed in one view. And so like many of the charts in Omicsoft, you can customize the volcano plot and color it based on expression. And so to do so, you can simply just go into the Task tab and select the option to change symbols properties. So here, when you click on Change Symbols Properties, you can customize it by selecting how to color your volcano plot. So in this case, go ahead and select continuous to do a continuous color. And using this drop-down, let's use the first graph as your anchor graph. So we're going to go ahead and color by the first comparison based on log 2 fold change. And so here you can see that this is your anchor graph, where the green represents downregulation and the red represents upregulation. And you can see that if you compare that with the coloring of the other graphs, right, you can see that some of the expression is very similar, where the genes that are downregulated in our anchor plot are also downregulated here, right, and here, right? But then you can also see that some of the genes that are upregulated in our anchor plot are actually downregulated in some of these other volcano plots. Right, so this allows you to take a look how similar or different that differential expression is compared to one of your volcano plots. Now you can also change how you want to label things, right? So in this case, you may not be familiar with the gene IDs. So let's go ahead and change it from gene ID to something much more simpler as a gene name. So once you make those changes, you can see that being displayed here in the volcano plot. And so this plot is interactive. So if you want to know, for example, what these down-regulated genes and what these upregulated genes are, you can simply just select them. So here, we'll go ahead and select some upregulated genes. And then also, if you click on Control on your keyboard, you can select additional genes, right? And so now you can see the names being displayed on what those individual data points are. And so when you select genes in your volcano plot, it is going to open up this table at the bottom with all of the information for those selected genes across your different comparisons that you are looking at. And so here, feel free to export this table into Excel or as a text file and use this information in downstream applications. Now, you can, in addition to exporting the table, you can also export your volcano plots. And so here you can export them in PowerPoint and present them as a slide, or you can also export them as a graphic by clicking on this little graphic icon. And so sort of reviews the steps that we've done so far, right? So we started off by launching OmicSoft and selecting your LAN by simply going to the LANs tab, using the select LAN and selecting your LAN of interest. You then search for your gene of interest under the search bar, selected it, and that brought up the default view as the default view of the comparison details versus in looking at normal and disease. So here, rather than looking at everything, what you did is you filtered for your disease of interest. So you went under the Projects tab and then expanded the project information section and selected the disease filter. Search for your disease of interest, which is in this case was Alzheimer's disease, and then clicked OK. That removed all of the comparisons, except for those focusing on Alzheimer's disease. And here you were able to visualize what those comparisons are. So each individual bubble represents a comparison coming from a public data set. You can easily select your comparisons that you're interested in by just drawing a rectangle. And what that's going to do is that's going to go ahead and open this table down here. That's going to provide you with information on sample level expression, what are some of the metadata that the project is associated with. And so if you want to take a look at the gene expression information associated with these comparisons, you can just select on Browse Selected Comparisons, and this will open up the volcano plots. This displaying differential expression for each of your comparisons of interest. You can change the layout to adjust it so that all of your plots display in one location. And you can customize it by going to the Task tab, selecting Change Symbols Properties. Here, doing a continuous color based on expression of a anchor plot. And then you can also change the name of the data points that appear on the volcano plot as well. So doing those customizations will allow you to take a look at the expression of one volcano plot and compare it to the others. And so here you can easily just select your data points and visualize them in the table that you see here. Now, today we focused on taking a look at disease versus normal comparisons, but you can also take a look at other types of comparisons as well, right? The default view will give you the normal versus disease, but if you go under the select view and go into the comparison section, you can see that there are different types of comparisons that you can check out. So if you are interested in taking a look at treatment versus control, you could just simply select that option, and that will display a bubble chart showing the comparisons for your gene of interest under disease versus control conditions. And so with this, let's go ahead and take our first Q&A break and answer any questions that came up.
Fabulous. Thank you so much, Araceli, for such a wonderful presentation so far. So a couple things for you guys before we take some questions. So first, I'm going to launch a poll just to check in with you guys to see is Araceli's speed looking good, and a few additional questions. I also would love to find out from you guys, based on what Araceli has gone through today, what are some of the metadata fields that you think you'd want to take advantage of when applying filters to look for some of this publicly available data? So if you could share some of that in the chat box, just pop in, again, ideas or maybe some of your favorite metadata that Araceli has shown, just so we can get a sense of what are the types of things that you guys are looking for when you're looking for these publicly available data sets. So again, that chat box is at the bottom of your Zoom interface screen. So if you could submit your ideas there, that would be wonderful. Also, additionally, I will be sharing a link to today's slides in that chat box after you guys are finished submitting your metadata top hits here. And let's see, it looks like we have a couple questions that have come through. One of them is in regards to the certification courses that we spoke about at the top of today's webinar. So the question was, can you clarify how folks can participate in those certifications since that registration deadline has already passed? And it looks like the answer to that, I don't know if you know, Araceli, that that link is still clickable.
I believe it's still clickable, but
Yep.
You mentioned earlier, if even though the deadline has passed, just reach out to us. We can see if we can still sign you up, or sign you up in our next round of certification.
Yeah, for sure, for sure. So, and also remember, it's a self-guiding or a self-learning curriculum. So if you do sign up for this course, even though that deadline's passed, you're going to have plenty of time to finish the course before that exam deadline, which is next year. So plenty of time to still try to join into the cycle for certifications at this time. Another question is, I'm not familiar with the lands, so where can I find more information about those lands?
Great question. So here, if you go under help and you take a look at our white paper release, our white paper release is going to provide you with information on what the lands have. So here, if you take a look at disease M, for example, it is going to open up a PDF in your computer where you can take a look at what are the different types of lands, what information they contain, and so forth. So the white papers are very useful to allow you to see what type of information each of them contains.
Awesome. Another question is, how often is OmicSoft updated?
So OmicSoft is updated on a quarterly basis. So every quarter we add more samples, more projects, more information to lands.
Excellent. Another remaining question that we have is, I know that you have grabbed these comparisons using a gene of interest, but what if you just wanted to look at the general comparison data without kind of targeting a gene of interest? How would you go about that?
So here, if you're interested in just looking at comparison data, you can simply just go back to the main tab, right? And you can change the way that we're viewing. So right now we're looking at the sample distribution, but if you go into select view, you can take a look at the comparisons. And so what this is going to do is going to generate the different comparisons based on tissue category. And so here you can see what those types of comparisons are. You can select them and browse in the same way that we did to generate these volcano plots.
That's awesome. All right, and just a quick reminder to you guys too, if you don't have access to Omicsoft Studio, the actual client, like I said at the top of the hour, all of this stuff that Araceli is doing, you could easily do within Land Explorer. So have a click around in Land Explorer, which can be accessed through IPA. And if you had any questions about that, or if you're lost with that, just reach out to us and we can have a one-on-one session to get you started with that. So in the meantime, I'm going to share again that link for today's slides in chat. And Araceli, you can take the reins back over.
Awesome. So now let's go ahead and take a look at how you can leverage public expression data. And so you can do this by generating heat maps and box plots to help you identify potential biomarkers and targets that you can investigate using HCMD and IPA as well. So now let's say that you read this GWAS study looking at potential markers and variants for Alzheimer's disease. Now in this study, they identified 9 genes that could potentially impact Alzheimer's disease. And so you want to go look at the expression of these genes to help you sort out whether these are good targets or perhaps they may be targets for something else. And so you can easily do that by simply going back to Omicsoft. So here, let's go ahead and go back to our tab. So if you go back to our comparisons tab, right, you can see that, as I mentioned, we're focusing on 2 different projects of interest, GSC 48350 and then GSC 5281. So today, just to simplify things, you'll focus on these 2 projects to help you look for potential biomarkers and targets. So how do we filter for just those projects? So if we go to our main tab, which displays all that information, you can filter for your projects of interest by simply going into the project tab right here. So here under the projects tab, you can expand the project information section, and you can search based on a session number by simply just clicking on the little filter icon. So here, you click on the little filter icon, you get to see all the different projects that are available within within human disease, right? So you can see that we have many, many different projects that you can search for. And so here we can simply just select our project of interest. So one of them is going to be, let's see, so it's 483. 48350. Here you go, 48350. And then our next project is going to be GSC 5281. All right here. So now that we have our 2 projects selected, you can just simply click OK. So this is one way to filter for your projects of interest. The other way is just to simply enter the filter that you're interested in, right? So if you don't want to search the way that I did, you can just simply click on the little filter icon and then just type in the actual filter that you want to apply. So now you'll only focus on the samples that are coming from those 2 specific projects of interest. So here, let's go ahead and search for your genes of interest. So to search for a single gene, we used the search bar. But to search for multiple genes, all you have to do is just go under Advanced and then select the option Search Multiple Genes. Here, you can go ahead and either type in or paste your genes of interest. I'm going to go ahead and just paste these in the chat box. So for those of you that are following along, you can join me as well. So I'm going to go ahead and just paste those, right? And so here you can see that we have 9 genes that you are going to be searching. So again, we went to the Advanced, clicked on Multiple Genes, and then typed in our genes. And so now we can go ahead and click OK. And so what this is going to do, this is going to search for the information for those multiple genes, only using the samples coming from our 2 projects of interest. And so similar to what we saw when we search for a single gene, when you search for multiple genes, the default view is going to be a heat map of the comparison analysis between disease states versus normal samples. So here this is just taking a little bit because it is searching for 9 genes rather than 1 gene. So while that goes on, just to kind of review what we've done, right? We went into our main tab. Here you went under the projects tab, under the project information, and then clicked on the little accession filter icon, searched for your studies of interest, and then clicked on OK. And so that I'm still thinking, okay, so that is going to generate a chart that's only going to be displaying the samples from that particular study. To search for your multiple genes, you then went into the advanced, search multiple genes, and then either copied or typed in those genes of interest and clicked OK. And so what that's going to do is that's going to generate this heat map. And so here, the heat map displays different genes on the y-axis and expression of those genes across different tissues on the x-axis. The red is going to represent regulation of that gene in disease versus normal, and then the green represents downregulation of that particular gene in disease versus normal. And so here, for example, you can see, as you saw earlier in my slides, that MED3 seems to be upregulated in Alzheimer's disease versus normal controls in these different tissues across the 2 projects that you selected. And so this is a quick way to identify genes that could be used as biomarkers or genes that could be broadly targeted across your different tissues. So let's go ahead and see if that has been loaded up. Oh, yes, here you go. Right, and so here you can see that we now have, again, our genes and the expression of those genes in disease versus normal here in this heat map. Now, the project that we are looking at today, it is a RNA-seq project, right? You're looking at microarray studies, but if you're interested in taking a look at the expression data for that project, you can simply just take a look at it very easily by just going into the expression heat map, right? Now, if you are interested in taking a look at RNA-seq data, then all the RNA-seq information is going to be found within the RNA-seq quantification section, right? So microarray data views are under expression, and then RNA-seq data is going to be under the RNA-seq quantification. So to look at the individual expression, right, so rather than looking at the comparisons, you may want to take a look at the individual expression of these genes. You can generate a heat map of individual expression by just simply, again, changing the way that we're viewing the data. So you can go under Select View, and then here under the Expression section, select the option to view the heat map expression intensity. And so what this is going to do, this is going to display a heat map of differential of gene expression for all of the individual samples that we're working with. So let's see if that has been generated. Nope. Okay. So once that loads up, what you're going to see is you're going to get a heat map that is clustered based on distance with different genes on the y-axis and expression of those genes across different samples on the x-axis. And so here, if you remove the x-axis dendrogram, you're able to sort of rearrange the way that these samples can be clustered, allowing you to cluster samples based on different disease categories, where in this case, the blue represents Alzheimer's disease samples, and then the green represents normal control samples. That's still going. So just to show hands, how many of you are familiar with using heat maps to take a look at expression information? So I can see, just go ahead, just raise your hand for that. So here I can see that some of you are interested in taking a look at expression information, right? So here you can easily see how genes are either upregulated or highly expressed, represented by the red, and then how some genes are have low expression, represented by the green. So in the meantime, Kristen, if there's any questions that I can just answer without showing anything, just let me know.
Sure. For sure. So a couple questions. One of them that came up while you were showing those filters that are over on the left-hand side, some of those guys were showing up as red fonts. What is the difference between those black fonts and the red fonts on the filters? Yeah.
Great question. So when you take a look at your left panel, the red font just represents that the filter was used. So in this case, you used the session filter to filter for particular studies. And so this is just kind of like a nice way to let you know, hey, this filter is being used compared to these filters which have not been used. And so that's what that is what the red font represents. Just you know, this filter has been applied in some form to the data that you're looking at. Oh, awesome. Okay, here we go. So here, as I mentioned, it's going to generate a heat map that it is clustered based on distance. And so that makes, you know, the arrangement of the samples a little messy. So one of the things that you can do is you can go into the Task tab, and then here select the option to hide x-axis dendrogram. And so when you do that, it's just going to help just the samples get rearranged a little bit better. And so now you can clearly see which are your disease state samples, and then which are your control samples. Now, taking a look at this heat map, it is kind of messy, right? You can't clearly tell, you know, exactly how these individual samples are expressed across, how these individual genes are expressed across these different samples. And so for that, you can generate box plots instead. And so you can easily do that by simply just going to Select View. And then here under the Select View option, within the expression section, you can use the option to generate a multi-gene variable expression intensity view. So when you go again to Select View, Expression, and then multi-gene variable expression intensity view, what this is going to do is it's going to generate box plots where you have genes on the y-axis and expression of those genes across different samples on the x-axis. Now you can compare the genes, right? But the samples are all clustered together. So you can split this plot into multiple plots. How to do so? By simply just going into the Task tab. So here, under the Task tab, what we're going to do first is we're going to go ahead and subgroup our information based on our disease category. So you can do that by just simply going into the Specify Profile columns. And so here, let's go ahead and select Group. And so again, your group is based on the grouping that you have selected. And so you can always change this if you like. In this case, selecting the group and moving it to the right-hand side will add that information. Click OK. And so now you can see that you have your genes, and then they are subgrouped between normal samples and disease state samples. So now that you can see how your individual gene is subgrouped into normal and disease samples, you can divide this into individual plots by, again, going into the task tab, and then here under the trellis section, using the option to trellis by covariate. So when you click on this option, you can split this one chart into many charts, in this case by using the gene name, moving it to the right-hand side and clicking OK. And so now you can see that you have 9 individual charts. Let's go ahead and just change the layout so that they are all displayed in one view by simply selecting the 3x3 view. And so here you can see that we have a individual chart for each of your genes of interest, and you can see how those samples, you can see the expression of those samples in the normal and in the disease state. Now, some of you may be interested in statistics, want to know what the significance in terms of the difference between these two groups is for each gene. And so for that, you can go under the Task tab, scroll down to the bottom, and under Customized, you can use the option to show summary information. So when you do that, it is going to provide you with some statistics, which you can then use to sort by p-value under the trellis section. So here, let's go ahead and just sort by p-value. And so this is going to prioritize those that are more significant at the top. So there's different ways, so you can always just hide the summary information, and you can take a look at those charts this way, right? So it's really not a surprise that MED13, which you saw clearly in the heat maps, seems to be one of the highly expressed genes in Alzheimer's disease samples compared to normal controls. And so this is a nice way to clearly see what some of your markers of interest could be, right? And so just to kind of review what we did, right? So we changed the way that we viewed the information from a heat map to individual box plots by going to Select View and then selecting the option of multiple gene variable expression intensity. Once you did that, the first thing that we did is do some subgrouping. So you went to the Task tab, selected the specified profile columns, subgroup based on the grouping, which in this case was a disease category, moved it to the right-hand side and clicked OK. And so once you did that, you then split the chart into multiple charts by going into the trellis by row covariate, selecting gene name to be the splitting factor, moving it to the right-hand side and clicking OK. And so that generated individual plots for your genes, which you can then show some summary information and sort by p-value, as you see here. And so with this, let's go ahead and go into our next topic, right? So after identifying your potential gene and biomarkers using public data available in Genomics, users like you are interested in variants. So let's go ahead and take a look at how you can use the Human Gene Mutation Database, HGMD. So as a use case, we're going to be focusing on this particular gene, GSN. So GSN was identified by this paper to be a marker for the disease-associated astrocytes in Alzheimer's. Now, it's also one of the 9 risk-associated genes for Alzheimer's disease based on this GWAS paper. And so what you want to do is you want to know if there are any variants in GSN that may be interesting. And so what is the Human Gene Mutation Database? And so it is an expertly curated database of human germline mutations coming from peer-reviewed published journals. These are going to be both de novo and inherited mutations that have significant evidence of pathogenicity. So if it's not published, it is not going to be an HGMD. Now, one thing to note is that HGMD is for germline mutations only, not for somatic. So HGMD is a great tool when you're asking the question, does this human germline mutation cause disease? And like with OmicSoft, it is expertly curated for the highest quality and consistency. And so HGMD can be accessed online. So let's go ahead and show you how you can do so. So when you log into HGMD, it is going to have an interface that looks like this, right? So here you'll just have some key information in terms of what's available within the database. Now here on the top, you can search by gene, by mutation, by phenotype to find information of interest. But you can also do what we call a quick gene search. So let's go ahead and use that option. So here under the quick gene, let's go ahead and type in GSN and search. And so when you do that, it is going to bring up any relevant gene information. So in this case, we can see that our gene is highlighted right here. And so let's go ahead and click on that option. So this is going to bring up all the information HGMD has on your gene, GSN. And so here, for example, you can see that there are different, it's going to show you the different types of mutations that are associated with that gene, and it's going to be split based on different types. So if you are interested, for example, in taking a look at missense mutations, you can go ahead and select that specific mutation type that you're interested in and filter things that way. Now, I just realized I just searched for the wrong gene. So let's go ahead and switch that. GNS. There we go. My bad. So here you can see again the different types of mutations that are associated with this gene, right? And you can also identify different types of mutations based on a variant class, right? So HGMD has its own classification system where it can classify something as disease-causing or possibly disease-causing. And so let's say that, you're interested in identifying all of the mutations that are disease-causing within your gene of interest, that you identify the publications and through your OmicSoft queries. You can just simply select on this option, get all DM, in this case, get all disease-causing mutations. And so what this is going to do is going to bring up a list of the different mutations that are known to be disease-causing, and it's going to be divided into different mutation types, right? So you can take a look at what the different types of mutations are for this particular gene that are disease-causing. Now here, you can easily take a look at this table to see what is the associated literature for those mutations that have been identified. So you can always go back to the literature and see how they identify the variant. You also get information on the codon and amino acid changes, as well as nomenclature information and what is the associated phenotype for that particular variant. Now, clicking on the accession number is going to provide you with more detailed information about this particular variant of interest. And so here we can see that the particular phenotype that this variant is associated with, what it is classified as. And it's also going to provide you information on some of the biochemical properties of this particular variant, right? So here you can take a look at the amino acid comparison. What is the influence that, changing from a serine to isoleucine is going to have in terms of the effect of the protein. And it also gives you information on the different predictions that are going to be predicted to results based on the mutation, whether this is going to be a deleterious mutation or a benign deletion as well, right? And so by taking a look at these different informations, it can help you determine whether this particular mutation is deleterious or not, or whether you can use it as a drug target. And so just to kind of review how we got here, right? So you search for your gene of interest, selected it, and then took a look at specific types of mutations. In this case, took a look at disease-causing mutations. But for those of you that are new and may be confused as to what these abbreviations are, this slide does include the original definition that the authors gave for the original paper, so you can always refer to that for insight. Now, once you select for your gene, for your different mutation types of interest, they're going to be displayed as a table, allowing you to quickly view the different types of information that those variants are associated with. Here, you can select the accession number and get more greater details on exactly what that variant is, and take a look at some of its biochemical and prediction information. Now, one thing to note is that in HGMD, not only do you get mutations, but you also get phenotypes. So for example, this mutation that we took a look at, which is this one right here, it is associated with a particular phenotype that you may not be familiar with. It kind of looks like it's a metabolic disorder. And so what you can do is you can search for this phenotype that's associated with, and see if it's associated with your disease of interest, which in this case is Alzheimer's, right? And so by taking a look at a segment from this paper, which is highlighted right here, you can see that indeed it seems that a subset of this condition or the classification of this particular phenotype is related to dementia in Alzheimer's disease. And so not only did you identify a variant, but you also identified a phenotype that may be associated with your disease state of interest. And so with this, we're just going to go ahead and take a quick Q&A break just to make sure that there's no remaining questions.
Fabulous. Thank you so much, Araceli. Good stuff. Couple questions that we had pop up is, how often is HGMD updated?
So HGMD is updated on a quarterly basis. So every quarter, you'll get a release of a new information. And you can always get that information here. If you go under statistics, it will display what was updated from the previous version. So here you can see the updates in terms of the content. Excellent,
excellent. How would a person go about searching for a very specific mutation in HGMD?
If you're interested in a specific mutation, you can just simply go into the mutation section. All right, and so here you can search for a specific mutation, either based on the codon that you're looking for, the actual accession number, the nomenclature, or the chromosomal coordinate. So you can enter any of these parameters here in the search bar, and then just select that type of parameter that you're searching for, and this is going to bring up the specific mutation that you are interested in.
Very cool. That looks like a pretty easy to use search tool here. All right, so I'm going to hand it back over to you. There are no other outstanding questions. And once again, I will share today's slides in the chat box one more time in case you guys have missed it, and take it back over, Araceli.
Awesome. Thank you, everyone. Please continue to type in those Q&A questions in the Q&A box. All right, so let's go ahead and take a look at how you can use IPA to discover potential mechanisms. So in IPA, not only can you build a network for your gene and disease of interest, but you can also use the QIAGEN knowledge base to see whether a gene is involved in a particular pathway and to start to explore that as well. So IPA is a powerful relationship discovery tool. So for more than 20 years, QIAGEN's team of MD and PhD level scientists have gone through the literature in top journals and defined the key molecular interactions found within those publications and pull out appropriate context for these relationships from the study to understand the interactions between molecules and molecules and diseases, association between the disease and the molecules, right? And so overall, the QIAGEN knowledge base represents a massive ontology of over 13 million curated findings, updated weekly and quarterly, that try to define molecular interactions and associations from literature, allowing you as the user to use decades of accumulated knowledge to map these different types of networks that you may be interested in. Now, in OmicsSoft, as we saw, you can search across different diseases and tissues, find comparisons, identify potential biomarkers, drug targets at the sample level. Now, after you have used the sample level information, you can then use IPA to highlight key pathways, to build networks, discover potential mechanisms of action. So you can use IPA and OmicsSoft back and forth to help you discover potential future molecules of interest. So let's say, let you know how we've used GNS as our example. We know that it is associated with Alzheimer's disease. We also know that, there are some variants that could be important in other conditions that have been associated or have sort of been linked to Alzheimer's disease. And so now you want to use IPA to know how is GNS and Alzheimer's connected. And so in IPA, you're going to build a network and use activity prediction to take a look at what happens to the network and the disease of interest when targets are either upregulated, represented by the red coloring, or downregulated, represented by the green coloring. So let's go ahead and see how you can leverage IPA. So when you open IPA, this is what the GUI interface is going to look like. Here on the left-hand side, we have what we call the Project Manager window that's going to store all of your data. The Create New gives you different access to features like uploading a data set or starting a core analysis. And then the search bar up here is going to give you access to leverage IPA's knowledge base to search for a gene of interest under the Genes and Chemicals tab, or search for a disease of interest under the Disease and Functions tab and get more information about that gene or disease. And so for today, you want to know how GNS, the gene you identified through the paper in HGMD, plays a role in Alzheimer's disease. And so for that, we're going to go ahead and be using the search bar. So under the Genes and Chemicals tab, you can type in the gene of interest, GNS, and as you do so, it is going to auto-populate. So you can go ahead and select it. So here, you can add this to a blank pathway to start your network construction by simply just selecting that gene and then add to my pathway. So once you select it, add to my pathway, you can go ahead and add it to a new pathway. And so this is going to open up a blank canvas where you now have your gene of interest. So now that you have your gene, you can search for your disease of interest by simply going into Diseases and Functions tab under the search bar. So here, go ahead and type in. So we'll start to type in Alzheimer's disease. And as you do so, it is going to populate. So you can select your disease of interest. And similar to what you did with the gene, right, you can select your disease. So in this case, whether you're looking at Alzheimer's disease from a psychological disorder standpoint or from a metabolic disease standpoint, the same number of molecules are associated with it. So just go ahead and select this one just at the bottom. And what you're going to do is you're going to go ahead and add it to my pathway. In this case, add it to that pathway that you have open. So this little window is going to ask you what type of information do you want to add. Do you want to add all of the molecules that are associated with it? Do you just want to add a disease or function as a node, or do you want to add both? In this case, you're only interested in disease or function as a node, so you can go ahead and click OK. So once you do so, you now have your gene and your disease. And so with this, you can start to use the different menus that are available to add information and build your path, your network. So the option that you're going to use is going to be the build menu, because again, we're going to build a network. So under the Dropbox, you can use the option which is called Path Explorer. So Path Explorer allows you to identify paths in which your two nodes, in this case, GSN and Alzheimer's disease, are connected by using this box here on the left-hand side. So in this case, you are interested in seeing how GSN connects to Alzheimer's disease. And so what we're going to do is you're going to add GSN to this set A box and Alzheimer's disease to this set B box. Right, so this is going to convert this into this is going to be sorry, I'm having issues with my annotations. This is going to be A, and then this is going to be B. So let's go ahead and select, add to set A, and then select, add to set B. And then in terms of the direction, again, we're interested in going from GNS to Alzheimer's. And so you're going to select from set A to set B. So once you've established how you want to generate this network, you can go ahead and click Apply. So this is going to display the different ways in which GSN through intermediates connects to Alzheimer's disease. So here you can select them all and add them to your your pathway, right? And by simply doing that, you now have a network that shows you how your gene and disease are connected. Now, once you have the network connecting your gene to Alzheimer's disease, you can take advantage of the quality in IPA's database to see if the expression of any of these genes, let's say GNS, influences the expression of other genes within the network and the disease overall. And so for that, you can use the option called Overlay. And so within the Overlay menu, the second option is what we call the Molecular Activity Predictor. And so here, what you can do is you can go ahead and use these color buckets to let IPA know how you want to change the expression of this or the activity of this network, right? So let's say that you want to know what happens when your gene of interest is upregulated. You can select the red bucket and color it red, right? And so you can see that based on the literature information, there's not enough evidence to support that the upregulation of GNS is having an influence on this isolated network, right? And so here you can always verify, the information that's associated within this network that's making these connections by just simply double-clicking on the connecting line, right? So here there is a protein-protein interaction relationship between GNS and this gene. And so you can get a little summary view as to, what that particular relationship is. Now, some of you may be looking at a gene for the very first time and may not know what that gene actually is. And so for that, you can just double click on the gene and it's going to open up this panel again on the right hand side that's going to provide you with information on what this gene is all about, right? So here you can see a little summary of what the gene is. You can get information on what type of complexes or what type of memberships this gene is involved in. And then you can also get information on the different types of pathways that this gene can be found in, right? And so all of the relationships, in IPA have findings associated with them. And so here, let's say, for example, you are interested in this clear pathway, right? And you want to know whether this pathway is either increased or decreased at the sample level. And so one of the things that you can do is you can actually go into Omicsoft. I'm just going to do this through slides just to kind of show you how you can do that. You can go into Omicsoft and you can go under the search, the advanced option. And rather than searching for multiple genes, you can actually search for pathways, right? And so when you do that, you're going to get this window that allows you to search for different types of pathways. And so since IPA and Omicsoft are connected, you can go and take a look at those QIAGEN signaling pathways and search for your pathway of interest. This is going to display some information on what that pathway is. If you're not familiar with that pathway, you can go ahead and read this. And it's also going to let you know how many genes belong to that pathway and what those genes are. So here, when you click on OK, what this is going to do is this is going to query the expression of all of these genes. In this case, it is going to query it, if we've filtered, if we focus on our 2 projects, it's going to query on these 2 projects that we filter for. And so that's going to generate a heat map, just like we did when we searched for those 9 genes. And so here you can always change the way that you're viewing things. And rather than looking at the heat map, you may want to look at the box plots. So you can do that by going to gene, multiple gene variable view, and then generating, those box plots for each individual gene in this pathway. You can then, sort them by p-value and identify what genes within this pathway are upregulated in our disease samples versus our normal control samples, really allowing you to take a look at how you can see whether this pathway is either increasing or decreasing at the sample level. And so this is the great way to identify potential biomarkers or targets for particular signaling pathways that you may be interested in. And so, as I mentioned today, we're working on the GUI, right? But there's different ways that you can access Omicsoft, HMD, and IPA data. So let's go ahead and take a look at those. So today you saw how you can use the GUI in Omicsoft and IPA and the web to search and identify potential biomarkers and targets, but there are different ways that you can access the data. So for Omicsoft, you can download the flat files that you can then integrate into your internal databases or into your current pipelines. And so there's also a API query engine for those of you who want to do specific queries using Python, R, or REST API. Now, for HCMD, we did use the web interface, but you can also download the data that makes up the database and build your own MySQL database for you to access all that variant information. And then the QIAGEN knowledge base, which is what is powering IPA, can be downloaded as flat files so that you can integrate them into your pipeline. There is also a API query engine that allows you to query specific data using R Python. And then there's also a knowledge graph representation that is supported by Neo4j. So if you are interested about these different ways of accessing the data, feel free to check out this on-demand webinar for curated knowledge graphs and validating experimental data sets. So today our focus was on the GUI, but here are some examples of the queries that you can do with Omicsoft, right? So you can take a look at projects that include samples with matching criteria. So you can do a query like this to pull out all the projects with Alzheimer's disease. You can take a look at which genes are frequently downregulated, upregulated in certain tissues, certain criteria. And then you can take a look at how you can take a look at which genes are frequently upregulated, downregulated, or how genes are differentially expressed between the different samples, and again, generate output for that information. And so for those of you that are interested in the information that makes up and powers IPA, here are some examples that you can do with the QIAGEN Biomedical knowledge base to answer questions such as, which molecules are associated with the disease. Again, you can use the metadata and the categories to find that information. You can answer questions such as which genes cause activation of disease state, or which pathways are known for a disease. So again, this gives you the flexibility to use and customize your own queries and dig up some information that way. So again, feel free to reach out to us if you are curious about any of these additional ways in which you can use and access the data from Omicsoft, HMD, and IPA. And so with this, thank you everyone for your time and for your questions. And if there's any remaining questions, I'd be happy to take them.
Wonderful. Thank you so much, Araceli, for such a great presentation here. So just a few questions here. How often is IPA updated?
So IPA is updated on a weekly basis, but it's also updated on a quarterly basis. So during our quarter updates, we get huge increase of information in terms of like public data sets, comparisons, and stuff like that. That's coming from Omicsoft into IPA.
Excellent, excellent. If users are only interested in a single gene, can they still build a network in IPA?
Yes. So right now, so today we used under the build menu, we used the Path Explorer option, right? Because we did have some nodes that we wanted to connect. But if you are starting out with a single gene, you can grow your network. And so here under the grow option, you can select your gene, select how you want to grow it, grow this network, and then just going to Livingston and apply that information. And what that's going to do is going to query the knowledge base and identify, in this case, what are some of the pathways that this gene is associated with, as well as some of the genes that it's associated with. So it allows you to expand with, just one gene as a starting point.
Fabulous, awesome. I don't see any other questions in the chat box or in the Q&A box. I did share a link to share for you guys to share your feedback for today's presentation in the chat box. So if you can just share your thoughts on that SurveyMonkey link. I'll also share once again the slides for today if you want a copy of them yourself. And be sure to visit the link that you signed up for this webinar to view it on demand at a later time here. So with that, again, we don't see any more questions in chat. So we'll stay on the line just for a couple more minutes to see if there's any latecomers to those questions, and hope to see you in future webinars.
Thank you, everyone, for taking the time to participate in today's session.
QIAGEN.
Sample to insight.